Hello and welcome to my series on the rapid review of the organelles of the cell. Today I'm going to be talking about the endomembrane system, and the components of the endomembrane system are the endoplasmic reticulum, the Golgi apparatus, and the various vesicles that are made by that Golgi apparatus, specifically the lysosomes and then the microbodies that are inside the cell. So here on the slide we see a picture of the rough endoplasmic reticulum, which is a large, hollow, flattened membrane that has sort of tubes or inside segments here, a lumen inside, called saccules or cisternae. And into that space, we talked about in the video on the nucleus and the nucleolus and the ribosomes, how the ribosomes that are bound to this membrane will actually produce their proteins into the inside of this system that then get changed by enzymes that are located in the system. One of the changes we talked about is that the signal peptidase actually cuts off a sequence of amino acids that signal to have this ribosome bind to the rough endoplasmic reticulum, but then they're modified in other ways and ultimately concentrated and either transported by vesicle or used as part of the rough endoplasmic reticulum later on for use in things like membranes. So the endoplasmic reticulum comes in rough and smooth varieties. We'll talk about the smooth kind a little bit later. So here we are, another view of the endoplasmic reticulum. You can see that it either comes in large, hollow, sheet-like forms or in tube shapes. Here's a view of an electron micrograph. We've been looking at sort of cartoon book drawings, and we've sliced through it. We can see it on edge here, and this would be the inside portions of it, and then you can see the ribosomes, the black dots on the outside. There again, another view. Here's the inside of the endoplasmic reticulum, and then the outside with the ribosomes. Now, showed you this picture before. The endoplasmic reticulum actually will bud vesicles off of it and send those vesicles of protein product onto the Golgi apparatus. The Golgi apparatus receives all of the vesicles on the cis side, CIS, cis side, and then through several layers of sort of flattened sac-like material that are filled with various enzymes that modify these products. They modify what's necessary, they concentrate it, and they package it within the Golgi apparatus, and then it either becomes small organelles for use within the cell, such as lysosomes and microbodies, or becomes a vesicle that is transported to the surface of the cell for secretion. So you have various secretion pathways that come out of the cell as well. Now, we can even package up material that's taken from the extracellular space and put it into the Golgi for processing as well. So this is a multifunctioning packaging plant, this Golgi apparatus, also part of the membrane system. Now, the interesting thing about these cell membranes is that they have an inside and they have an outside. And within the lumen of these vesicles, or say Golgi apparatus, or say the rough endoplasmic reticulum, we've colored the inside phospholipid layer blue and the outside phospholipid layer brown. Just like you have a shirt, it's got an inside and an outside. But watch what happens. Here the inside's in, and the inside's in, and the inside's in, and when it joins up with a new sac, let's say within the Golgi apparatus, going from one layer to the next, the inside still joins with the inside. So that we're maintaining sort of a regularity this way. However, when we open these vesicles to the outside, whatever was on the inside turns inside out. And so this is one of the ways we actually add to the cell membrane itself, is by packaging up cell membrane within the Golgi apparatus and then exporting it via a transport vesicle to the plasma membrane of the cell. Here's another view of the Golgi apparatus. Here's my endoplasmic reticulum. And keep in mind that there can be both a smooth endoplasmic reticulum and a rough endoplasmic reticulum. Well, on the cis face, the receiving face of the Golgi apparatus, these little vesicles get received, concentrated, packaged, and then actually are exported off the transphase. Here's a view of the Golgi underneath electron microscope. Then lysosomes are little bags full of digestive enzymes, hydrolytic enzymes, that are used either to digest old components, old organelles within the cell, or can be used to digest an intruder. In the case of a white blood cell that uses phagocytosis to engulf a bacterial intruder and then would join that little vesicle it's made up with a lysosome and then digest the little bacterial cell. Or they can be used for programmed cell death. Certain cells have to die at certain times for the good of the organism. For example, the tail of a tadpole disappears because of the apoptosis, the programmed cell death of these lysosomes bursting open and digesting the cells in which they'd lived. We also have some smaller bodies that are not part of the endomembrane system that are nonetheless produced as products of these concentrated Golgi vesicles, the peroxisomes and glycosomes, for example. Smooth endoplasmic reticulum makes lipids, so steroids and fats, whereas rough endoplasmic reticulum makes proteins, but both send their products to the Golgi to be packed. Now that finishes our discussion of the endomembrane system. Feel free to tune in next time when we'll be discussing non-endomembrane system organelles and energy-related organelles such as the mitochondria and the chloroplasts. Thanks for watching.